So we are going to begin this session. Welcome all to the real science. And this session, as you know, this first plenary session is a mixture of the two of the main neurodegenerations we deal with. The first one will be on Parkinson's disease. It is a pleasure and privilege to introduce Professor Anthony Schapira. He's from Manchester, and a Manchester United strong supporter, but he has made most of his professional work neurologically wise in London. He's the professor and chairman and dean of the Royal Free Medical School campus in the west of London. And he's very well known for his major contributions to the etiopathogenesis of Parkinson's disease. Like 30 years ago, he started the thinking about Parkinson's disease as a failure of energy production by describing the very initial reduction in cytochrome oxidase 1 and 3 of the mitochondrial change in the Nigra of PD patients, and thereon has made a large number of contributions, both by examining patients and experimental work. We have done work together. And in the last few years, and the topic of today's presentation, he has played a leading and fundamental role in describing and unraveling the role of the GBA enzyme as a mechanism underlying the neurodegeneration process of PD, and no doubt today he will further illuminate us with current work. Thanks a lot, Tony, for coming, and pleasure to have you here.
Thank you very much, Jose, and for that very kind introduction, and thank you also to the organizers, to Miguel, for their very kind invitation to come and speak to you today. What I thought I would do this afternoon in the time available is talk to you across the spectrum of the link between glucocerebrosidase mutations, or I'm going to refer to that as GBA for short, and the development of Parkinson's disease. Some of you, of course, may be very familiar with this area, and I apologize if I give you some information you already know. But those perhaps with an interest more on Alzheimer's disease might find this topic new and of interest. The first slide is really an overview, and it would work very well. There we go. Is an overview of the topic of the link of GBA and Parkinson's disease. I'm then going to compare and contrast those patients with Parkinson's disease who have with those who do not have GBA mutations, to ask, is there a difference in the type of Parkinson's disease? I'll then talk to you about the biochemical abnormalities induced by GBA mutations and how those abnormalities may contribute to the development of Parkinson's disease. Then I'm going to move on to discuss the GBA pathway as a potential target for Parkinson's disease. This pathway is not going to be a target for symptomatic therapy. This pathway will be a target for neuroprotection or disease modification therapy. And I'll show you the data that we and some others have had from some models in this area. And then finally, I'm going to briefly touch on the clinical trial that's now about to be started. It's received approval, and the first patient will enter later this month, testing a compound that is targeted to the GBA pathway to help determine whether this will be of value in slowing down Parkinson's disease in the future. So that's what we're going to talk about over the next half an hour or so. So, thank you. The genetic causes of Parkinson's disease amount currently for a relatively small minority of cases until the GBA gene risk association with Parkinson's disease. The single gene mutations probably accounted for less than 5% of Parkinson's disease. In some communities, of course, that figure was much higher, usually due to a founder effect. The more recent GWAS studies have identified a number of association genes with Parkinson's disease, and there is a general belief that much of Parkinson's disease is genetic, but complex genetic, not single gene. However, the single most important genetic factor for Parkinson's disease is GBA and GBA mutations. The LRRK2 mutations, previously thought to be the most common, accounted for about 0.5% of sporadic Parkinson's disease. GBA mutations are found in anywhere between 10 and 25% of patients with Parkinson's disease. In the UK general PD population, the figure is 12%. So this association of GBA with Parkinson's disease is by far the most important genetic association. Just to remind you of where the GBA pathway fits in the biochemistry of the cell, it is a crucial part of ceramide metabolism. Mutations of the GBA gene have been known about for many decades, associated in homozygous form with Gaucher disease, a lysosomal storage disorder in which there is accumulation in lysosomes within multiple organs associated with some interesting features such as increased risk for cancer, myeloma, osteoporosis, etc. And Gaucher disease exists in itself in three different forms, from relatively mild to quite severe neuronopathic forms. There are over 300 different mutations of the GBA gene associated with Gaucher disease. So this enzyme fits in a pathway important to cellular metabolism, and you can see from this slide those enzymes marked with a red star are those associated with neurological disease when mutated. So you can see that this pathway itself is rich in the potential for causing neurological damage. So let me come back to a few more specifics about the GBA gene. I'm going to call it GBA rather than GBA1. There are two types of GBA gene, but it's number one which is the more important. So, of course, there's a homozygous and heterozygous state. This is an autosomal recessive gene mutation. Homozygous causes Gaucher disease. Heterozygous, previously thought to be completely asymptomatic. But it was the interesting clinical observations that individuals with Gaucher disease and their asymptomatic carrier siblings and parents had a much higher risk of developing Parkinson's disease. Not everybody with a GBA mutation gets Parkinson's disease. In fact, a minority, about 30% get Parkinson's disease. So if you have a GBA mutation in heterozygous form, it does not mean you'll get Parkinson's disease, but you will be at risk. And there has been a debate whether this is a risk gene or whether it's a dominant gene with variable penetrance. I think the simplest way to look at it is that it's a very important, potent risk factor. So 30% of carriers will get Parkinson's disease. For the individual, a GBA mutation leads to an increased risk of Parkinson's disease of 20 to 30 times. To put that in perspective, one of the oldest, in this case, environmental associations with Parkinson's disease was exposure to pesticide. And that risk factor was increasing Parkinson's disease by 2 to 3 times. So you can see that a 20 to 30-fold increased risk is really a major contributor to the disease. And as I've mentioned, 10 to 25% of patients with Parkinson's disease are found to have GBA mutations. That number varies according to whether you do complete sequencing of the gene or you only look for the common mutations. It also depends upon the ethnic origins of your target population. GBA mutations are much more common in the Ashkenazi community. So if you have a lot of Ashkenazim amongst your sample, that will lead to a falsely high level. But in the general UK population, the figure is of Parkinson's disease is 12%. Perhaps just as interestingly, or maybe even more interestingly, GBA mutations are associated with a very significant increased risk for dementia with Lewy bodies. In fact, the risk is probably greater for those individuals who carry GBA mutations to develop dementia with Lewy bodies. So there's the background. What about the clinical aspects? Are patients with GBA mutations who develop Parkinson's disease any different from the regular Parkinson patient? And the answer is actually not very much. You cannot tell the individual in front of you with Parkinson's disease whether they have or whether they do not have a mutation. Those with the mutations have slightly earlier onset, maybe about 4 to 5 years earlier than the general Parkinson population. They tend to have more cognitive impairment over time, and that cognitive impairment probably develops more rapidly. The pathology at post-mortem is identical. Lewy body deposition, neurite formation, etc. There is no difference in the pathology. The pharmacology is the same. The patients with GBA mutations respond in exactly the same way. They get dyskinesia, and in fact at the Queen Square DBS clinic, 17% of individuals were found to have GBA mutations. Imaging is identical to idiopathic Parkinson's disease, particularly in terms of PET and SPECT DAT scan. So asymmetric posterior putaminal loss of dopamine uptake. Interestingly different from other autosomal recessive causes of Parkinson's disease, such as Pink and Parkin, which tend to have a more symmetrical pattern. So, let me tell you a little bit now about some of the work that we've been doing over the last 5 to 6 years. We were very fortunate in London in having the National Lysosomal Storage Disease Centre only 1 floor away from the neurology service. So we were able to collect together over 100 individuals with GBA mutations who were completely normal clinically. They had no symptoms and on neurological examination had no signs. So these were an asymptomatic cohort of GBA positive individuals, either homozygous or heterozygous. But what we found was that even though these individuals appeared normal and their examination was normal, when you started to look at some of the biomarkers, the prodromal biomarkers of early Parkinson's disease, so you began to see some abnormalities in these apparently normal people with the mutations. So you'll see, for instance, here in the UPDRS Part 3, looking at subtle motor changes. There were abnormalities in the homozygous and the heterozygous individuals. Nothing that led to symptoms, but sufficient to be detected on examination. And then there was the smell test, the upset, abnormal in the individuals with GBA mutations. Not in everyone, of course. We're looking here at the means across groups. And I'll come back to the relevance of that later. And cognitive function was also slightly impaired, but significantly so, in those individuals with mutations. So we already began at baseline to see a difference between those with and those without mutations. Now, this is a longitudinal study, and at 2 years, we also began to see other abnormalities emerging, which are reminiscent of prodromal Parkinson's disease. So, sleep disorder, RBD, began to emerge as a significant problem in these individuals. None of them had Parkinson's disease, remember, at baseline or at this stage. And the Beck Depression Inventory also began to appear significantly abnormal among these individuals. The most recent data, which we've just completed, now brings this longitudinal cohort up to 5 years. And what we've seen across this cohort is a gradual progression of those early biomarker features. So the upset is deteriorating. There's also a deterioration in cognitive function as measured by the MoCA, in terms of the BDI, and also in terms of UPDRS 2 and 3. And what we're beginning to see here, in fact, when we look across the groups and start to put together some of these prodromal features, what we're seeing is that there appears to be a subgroup who are progressing more rapidly than others. So some of the GBA positive individuals are accumulating and progressing on these prodromal features more than other GBA mutation carriers. And in fact, using a mathematical model, you can take the cognitive function, the BDI, and the upset as a combination together and begin to separate out the so-called fast progressors. And when you do that, you can identify within the 100 GBA mutation individuals a proportion, somewhere between about 15 and 20%, who are progressing much more rapidly towards Parkinson's disease, as judged by these prodromal features, than the others. And indeed, some of our patients who were asymptomatic and completely normal 5 years ago have now developed Parkinson's disease. So what we have here is a longitudinal clinical cohort that's enabled us to identify some prodromal clinical features, which when we put them together enables us to identify those amongst the GBA mutation community who are more likely to develop Parkinson's disease than others. And the relevance of that will be apparent shortly. What about the biochemistry of GBA? Well, we looked at the brains of the Queen Square Brain Bank that had been identified to have GBA mutations. And of course, not surprisingly, those brains with mutations had lower enzyme activity of glucocerebrosidase. And this was generally throughout the brain, but most severe in the nigra and the striatum. That deficiency, of course, is not particularly surprising. What was surprising was that there was also a deficiency in the nigra in idiopathic Parkinson's disease without GBA mutations. Now, that was a big surprise to us. Why should GBA activity be reduced in individuals without a GBA mutation? You can also see the GBA deficiency in terms of enzyme activity in the blood in individuals who carry mutations. This is work from Roy Alcalay in New York, who showed that amongst the Parkinson's disease community, those with the mutations had lower enzyme activity in blood. So you could diagnose a mutation carrier, if you will, from the blood. Although the separation you'll see has overlap, so you cannot avoid doing genetic analysis as well. What's the observation of reduced enzyme activity in the non-GBA Parkinson brain led to was this idea that there is a reciprocal association between the reduction in enzyme activity and alpha-synuclein deposition. The theory being that if you reduce GCase activity, you will increase synuclein. Or alternatively, if you increase synuclein, you'll reduce GCase activity. And I'll briefly show you some of the data that supports this hypothesis. So the first is quite simply, well, knock out GBA in mice. So you knock out the gene, and what happens in the mice is that they accumulate alpha-synuclein. So a complete knockout of the gene in these mice leads to significant increase in alpha-synuclein. Instead of knocking it out, if you knock down GBA in cells, and here we're using SH cells, you see a significant increase in the accumulation of alpha-synuclein. So knockout or knockdown leads to accumulation of alpha-synuclein. Instead of manipulating the genetics, if you instead inhibit the enzyme with something called CBE, so you inhibit the GBA enzyme activity, you get an accumulation of alpha-synuclein in the cells. So knockout, knockdown, or inhibition of the enzyme all leads to increased alpha-synuclein. What about the other way around? If you increase alpha-synuclein, do you reduce GCase activity? Well, you do. If you overexpress alpha-synuclein in cells, here SH cells again, you get a significant reduction in GBA enzyme activity. Perhaps more importantly, if you take dopaminergic iPS-derived neurons from Parkinson patients with alpha-synuclein triplication, which are overexpressing synuclein, you see a reduction in GBA activity. And also, if you take alpha-synuclein overexpressing mice, at 20 months, there's a small but significant reduction throughout the brain in GBA activity. So, again, all of the models used indicate that increasing synuclein reduces enzyme activity. And this is probably the explanation for the reduced enzyme activity in the idiopathic PD patients. So, what is the mechanism for this reciprocal relationship between GBA and synuclein? Well, we don't know, but there are many possible explanations which are not, of course, mutually exclusive. So there's lysosomal impairment because most of synuclein is metabolized through chaperone-mediated autophagy. So lysosomal impairment, and that can cover not only the reduced GBA activity, but the reformation of lysosomes, and so on. Lipid alterations. Alteration in the lipid constitution of cell membranes caused by impairment of GBA activity in the ceramide pathway may interact with synuclein's interaction itself with lipid. There's ER stress. The GBA mutations, in some cases, are misfolded and become stuck in the endoplasmic reticulum, causing ER stress and unfolded protein response. There's the potential for some direct interaction between synuclein and GBA, although the data on this is perhaps a little less convincing. The reduction in GBA activity in a cell and in the lysosomes leads to an increase in alpha-synuclein release through exosomal release at the cell membrane. So the potential therefore is provided for this spreading phenomenon to occur. So I'm now going to turn from the clinical and the biochemistry to the important topic of, well, how do we make use of this information to consider the therapeutic target? And it's that last part I was talking to you about, the reciprocal relationship between synuclein and GBA, which I think provides the clue. Because if it is true, and everything seems to suggest it is, that reducing GK's activity will increase synuclein, the hypothesis is therefore, if you increase GK's activity, will you reduce synuclein? So to answer that question, we and others around the world have developed a number of models to try and determine if the manipulation of the reciprocal pathway can indeed reduce synuclein levels. So Drosophila, mice, and human iPS, or adipose stem cell neural crest-derived cells. Before I show you the first model, let me tell you a little bit about some of the drugs that you're going to see to determine whether that target is correct and how we're manipulating this synuclein level. This is a cartoon showing you the function of a drug called ambroxol. Ambroxol is a GBA chaperone. So it has capacity to link with GBA, correctly fold it, and chaperone it from the ER to the lysosome, leading to increased GBA activity in the lysosome. The drug also has other activities too. It is probably a transcription factor for the lysosome pathway. These are the data from fibroblasts taken from individuals with the two most common GBA mutations, the N370S and the L444P, as well as from idiopathic Parkinson patients. And what you see in these groups of cases is here you see the untreated and then the ambroxol treated, and then another chaperone called isophagamine, and then a fourth chaperone whose identity must remain secret at present. But all of these molecules, which all have very similar function of chaperoning GBA, all increase the mutant enzyme activity in fibroblasts derived from patients. Not only that, but when you look at the wild type, you also see a significant increase in enzyme activity, as well as you do in the idiopathic non-GBA individuals. And the relevance of this, I'm sure you are beginning to see. The next is the Drosophila model. So the Drosophila model, Drosophila doesn't have a natural GBA enzyme, but what it does have is the ability to manipulate and knock in the GBA mutations. When you knock in the GBA mutations that are seen in humans into these flies, you see a significant and progressive decline in their climbing ability, and you see a loss of TH-positive neurons. So the introduction of these GBA mutations begins to introduce a motor deficit and loss of dopaminergic neurons. You also see in these cells, and this is the retina of the fly, that is a very good model for showing you ER stress. And you'll see that there is a significant increase in the production of ER stress in these flies where the GBA mutation is expressed, either the N370S or the L444P. So the introduction of these mutations into flies leads to loss of climbing ability, so motor deficit, loss of TH neurons, and increased ER stress. The use of the GBA chaperones, ambroxol and isophagamine, reverse those deficits. So you have a reversal of the climbing deficit towards an improvement of motor function. You have a protection of the TH-positive cells, so you see less loss of dopaminergic neurons. And you see an increase in GCase activity. These flies do not express synuclein, so you cannot ask the question, does the ambroxol manipulate the synuclein? But in this next model here, these are dopaminergic neurons derived from patient samples, either through fibroblasts, iPS cells, or through stem cells in the adipose neural crest tissue. What you see here is that in the middle, the individual cells with the GBA mutation have a significant reduction in GCase activity, as you would expect with a mutation. And the administration of the GBA chaperone ambroxol leads to a significant increase to normal of the GCase activity in these dopaminergic neurons, and a supranormal effect in the non-GBA cells. Importantly, in these cells, you can ask the question about the synuclein, and you'll see that in these cells, the introduction of the GBA chaperone leads to a significant reduction in the alpha-synuclein deposition without any change in the synuclein mRNA. So the GBA chaperones increase GCase activity and reduce synuclein in these iPS or ASC-derived cells. These are data taken now from the mouse model of Parkinson's disease, but I'm going to first of all show you the wild-type effect. So here we see the effect in different brain areas of different concentrations of the GBA chaperone ambroxol in normal wild-type mouse brain. And this shows that the chaperone is capable of increasing the enzyme activity throughout the brain in these normal mice, which is important not only that it tells you that it's working in the mice, but it crosses the blood-brain barrier, at least in the mice. But it does that not only in the wild type, but it does it in the L444 mutant GBA mice too. So you see a progressive increase of GCase activity in these mice. So the ambroxol works in the cell models, and it works in the animal models, at least in terms of increasing the enzyme activity. These are some of the synuclein overexpressing mice I showed you the data from a little while ago. In the brain tissue of these synuclein overexpressing mice, there is, of course, an increase in synuclein. Total and phosphorylated. The use of the GBA chaperone after a very relatively short exposure of only 10 days, leads to a significant reduction in the synuclein levels in these mice, in the brain. So the GBA chaperone is capable of increasing GCase activity and reducing the synuclein. And of course, this reduction in synuclein in these mice is paralleled by an increase in the GCase activity. So, to bring all of that together, the clinical cohorts, the biochemistry, and the models, and the therapeutic targeting, what are the potential advantages of the GBA pathway as a therapeutic target for neuroprotection in Parkinson's disease? Well, if in the human case, the synuclein is reduced as it has been in the cell and animal models by GBA chaperones, that will reduce, of course, the total content, the phosphorylated alpha-synuclein content, and decrease the potential for spread of synuclein from one neuronal area to another, a feature which is thought to underlie the spread of synuclein pathology in Parkinson's disease. So there may be multiple ways in which this can work towards reducing the progression of Parkinson's disease. So this work has all led to a clinical trial that we have just initiated in London, looking at patients with Parkinson's disease, with GBA mutations and without, 50-50. And they will take, in an open label fashion, ambroxol. A relatively high dose, but the drug is extremely safe. And the primary endpoints of this clinical trial are really biochemical. We want to be able to prove that the ambroxol gets into the brain in humans. There is, in fact, a study using ambroxol in type 3 Gaucher disease in a small handful of patients in Japan, which does show ambroxol penetrates the blood-brain barrier. But I feel that we need to confirm that in this age group of patients in Parkinson's disease. We'll also be looking at the biomarkers of GCase activity in the CSF, synuclein, etc. And if this small, relatively brief study is positive, then that will serve as a means to initiate a much larger clinical trial with a clinical endpoint, looking to see if this drug ambroxol is able to slow the progression of clinical Parkinson's disease. So on that note, I'd like to end, and thank you very much for your attention, and hope that at least part of the talk has been of some interest. Thank you very much.
Thanks a lot. This is British punctuality. Questions, please. Is there any questions or comments? We do have a few minutes.
I have a question over here.
Okay.
Could you comment on the mechanistic basis for the link between this pathway and synuclein? You had a slide where you showed how they may interact, but it wasn't clear to me if that has been documented biochemically or if those are hypotheses of how you think the two pathways are connected.
Yeah, so I think at this stage those mechanisms must remain hypothetical. Some of the studies have demonstrated interaction at the lipid membrane between GCase and synuclein. Some studies have shown co-immunoprecipitation of the two proteins, others have not. Other studies have shown that the GCase activity leads to a deficit of lysosomal reformation, increase in exosomal export of synuclein, and so on. So I suspect, in fact, that several of those potential mechanisms are at play rather than just one of them, and that there's a complex interaction between the two. But the definitive mechanisms have not been identified yet.
Dr. Lanciego?
Hi. I completely agree on the sense that increasing GBA activity may be very useful to produce a clearance of aggregated forms of synuclein. I would like to know your opinion about gene therapy approaches using the GBA1 gene in an attempt to increase GCA's activity.
Yeah, so rather than use molecular chaperones, but to manipulate the GBA gene itself, and this has actually been done in animal models by Pablo Sardi, who has introduced the GBA gene using AAV. And using AAV into the GBA mutant mouse that he has, he was able to reduce synuclein. Of course, with an increase in GBA activity. So there is a potential to increase activity of the enzyme to reduce synuclein via genetic manipulation. And there are some studies which suggest that things like HDAC inhibitors may increase GBA function as well through manipulation of gene function. So I think that there's certainly more than one potential. I think the gene therapy idea does have attractions. Of course, access to the CNS in humans is not always so easy, but that may be overcome in the future by different delivery techniques. The other thing to remember, the manipulation of the GBA pathway has advantage not only for the brain motor function of Parkinson's disease, but the non-motor aspects of Parkinson's disease, because the drugs potentially are systemically distributed and therefore will have access to the peripheral autonomic nervous system. And if by reducing synuclein deposition, which occurs very early, of course, in Parkinson's disease, may again slow or prevent the development of non-motor features. So I think genetic manipulation or chaperone manipulation still both are possible avenues.
I'll have a comment to the question. I would have thought a very fundamental question here is what is the difference between the GBA mutants who go away to develop or don't develop? What are you? I'm sure you are examining this.
Yes.
There must be some difference.
Yes, so the question, of course, is a very good one, very fundamental. Why do some why do some individuals with GBA mutations get Parkinson's disease and others not? In fact, only the 30% get. So, of course, we don't know the answer to that. There are many possible reasons that maybe are probably genetic.
Other genes, you mean?
Yes, other gene effects or somatic effects. One could argue there may be environmental influences. That's possible too. So I think that this is a very important question to answer. What we have started to do is to do the very detailed genetic analysis of the fast progressors versus the slow progressors to see if we'll find any differences.
I have a question regarding the AD-PD comparison. In the AD field, we start to see a lot of development with the antibodies towards the abnormal protein. How far are you with antibodies against alpha-synuclein? Is that a valid approach? Or?
Yes, I think things are not as far advanced as in Alzheimer's disease, but there are early clinical trials, phase 1, phase 2 clinical trials, looking at immunotherapy in Parkinson's disease targeting alpha-synuclein. I don't think there are any data to share other than at least the first clinical trial appeared to be safe. And a larger clinical trial looking at a clinical endpoint is now underway. What I would say is that I think in Parkinson's disease, we should seriously be thinking about multiple modes of therapeutic intervention. So the use of immunotherapy will not be exclusive to the use, for instance, of a GBA manipulator.
Thanks a lot. Thank you very much. I think we move to the next speaker.